Chapter 40, the lymphatic system. Coming to the formation of lymph, lymph is derived from interstitial fluid that flows into the lymphatics. Therefore, lymph as it first enters the terminal lymphatics has almost the same concentration as the composition of the interstitial fluid. The protein concentration in the interstitial fluid of most tissues averages about 2 grams per deciliter, and the protein concentration of lymph flowing from these tissues is near this value. Lymph formed in the liver has a protein concentration as high as 6 grams per deciliter, and lymph formed in the intestines has a protein concentration as high as 3 to 4 grams per deciliter. Because about 2/3 of all lymph normally is derived from the liver and the intestines, the thoracic duct lymph, which is a mixture of lymph from all areas of the body, usually has a protein concentration of 3 to 5 grams per deciliter. The lymphatic system is also one of the most major routes for the absorption of nutrients from the GIT, especially for absorption of virtually all fats in the food. So indeed, after a fatty meal, thoracic duct lymph sometimes contains as much as 1 to 2% of fat. Finally, even large particles such as bacteria can push their way between endothelial cells of lymphatic capillaries and in this way enter the lymph. As the lymph passes through these lymph nodes, the particles are almost entirely removed and destroyed. Coming to the rate of lymph flow, about 100 ml per hour of lymph flows through the thoracic duct of a resting human, and approximately another 20 ml flows into the circulation each hour through other channels, making a total estimated lymph flow about 120 ml per hour or 2 to 3 liters per day. Coming to the effect of interstitial fluid pressure on lymph flow, we have this figure that shows the effect of different levels of interstitial fluid pressure on lymph flow as measured in animals. So we need to note that normal lymph flow is very little at interstitial fluid pressures which are more negative than the normal value of -6 mm of Hg. As the pressure rises to 0 mm of Hg, that is atmospheric pressure, flow increases more than 20-fold. Okay, therefore any factor that increases interstitial fluid pressure also increases lymph flow if the lymph vessels are functioning normally. Such factors include elevated capillary hydrostatic pressure, because it is a pushing pressure, it pushes fluid out of the capillaries into the interstitium, and from the interstitium it again goes into the lymphatics. Decreased plasma colloidal osmotic pressure, which is a pulling force. As the pulling force decreases, your lymph flow increases. Increased interstitial fluid colloid osmotic pressure and increased permeability of the capillaries. All of these factors cause a balance of fluid exchange at the blood capillary membrane to favor fluid movement into the interstitium, thus increasing interstitial fluid volume, interstitial fluid pressure, and lymph flow all at the same time. However, you need to note that when the interstitial fluid exchange at the blood capillary membrane when the interstitial fluid pressure becomes 1 or 2 mm of Hg greater than the atmospheric pressure, as you can see here, lymph flow fails to rise any further at higher pressures. This results from the fact that increasing tissue pressure not only increases the entry of fluid into the lymphatic capillaries but also compresses the outside surfaces of the larger lymphatics, thus impeding the lymph flow. At higher pressures, these two factors balance each other almost exactly. That is, the atmospheric pressure, it balances the interstitial fluid pressure, and so lymph flow reaches a maximum lymph flow rate. So this is illustrated by the upper level plateau in a graph between the relative lymph flow and the interstitial fluid pressure. Lymphatic pump, it increases the lymph flow. Valves exist in all lymph channels, as you can see here. See the valves, they exist in all lymph channels. So when a collecting lymphatic or larger lymph vessel becomes stretched with fluid, the smooth muscle in the wall of the vessel automatically contracts. Furthermore, each segment of the lymph vessel between successive valves functions as a separate automatic pump. So that is, even slight filling of a segment causes it to contract, and the fluid is pumped through the next valve into the next lymphatic segment. So this repeats, and in a very large lymph vessel such as the thoracic duct, the lymphatic pump can generate pressures as high as 50 to 100 mm of Hg. Okay, this lymphatic pump contraction occurs till the lymph is emptied into the blood circulation. Coming to the pumping caused by external intermittent compression of the lymphatics, any external factor that intermittently compresses the lymph vessel can also cause pumping. In order of their importance, such factors are contraction of surrounding skeletal muscles, which might occur in movements of parts of the body, and also pulsations of arteries adjacent to the lymphatics, and also compression of the tissues by objects outside the body. The lymphatic pump becomes very active during exercise, often increasing lymph flow to about 10 to 30 fold. Conversely, during periods of rest, lymph flow is sluggish and almost zero. Coming to the lymphatic capillary pump, The terminal lymphatic capillary is also capable of producing lymph in addition to the pumping by larger lymph vessels. The walls of the lymphatic capillaries are tightly adherent to the surrounding tissue cells by means of their anchoring filaments. Therefore, each time excess fluid enters the tissue and causes the tissue to swell, the anchoring filaments pull on the wall of the lymphatic capillary and fluid flows into the terminal lymphatic capillary through the junctions between the endothelial cells. Then, when the tissue is compressed, the pressure inside the capillary increases and causes the overlapping edges of the endothelial cells to close like valves. So therefore, the pressure pushes the lymph forward into the collecting lymphatic instead of backward through the cell junctions. The lymphatic capillary endothelial cells also contain a few contractile actomyosin filaments. Now coming to summary of factors that determine lymph flow. Two primary factors determine lymph flow are present, and they are the interstitial fluid pressure and the activity of the lymphatic pump. So one can state that roughly the rate of lymph flow is determined by the product of interstitial fluid pressure times the activity of the lymphatic pump. Now, the lymphatic system, it plays a key role in controlling interstitial fluid protein concentration, volume and pressure. So it is all an intertwined mechanism. So it is clear that the lymphatic system functions as an overflow mechanism to return excess proteins and excess fluid volume from the tissue spaces into the circulation. Therefore, the lymphatic system also plays a central role in controlling the concentration of proteins in the interstitial fluids, the volume of interstitial fluid and the interstitial fluid pressure. So let's see how these factors interact. First, we need to remember that small amounts of proteins leak continuously out of the blood capillaries into the interstitium. Only minute amounts, if any, of the leaked proteins return to circulation by way of the venous ends of the blood capillaries. Therefore, these proteins tend to accumulate in the interstitial fluid, which in turn increases the colloid osmotic pressure of the interstitial fluids. Second, the increasing colloid osmotic pressure in the interstitial fluid shifts the balance of forces at the blood capillary membranes in favor of fluid filtration into the interstitium. Therefore, in effect, fluid is translocated osmotically outward through the capillary wall by the proteins and into the interstitium, thus increasing both interstitial fluid volume and interstitial fluid pressure. Third, the increasing interstitial fluid pressure greatly increases the rate of lymph flow, which in turn carries away the excess interstitial fluid volume and excess protein that has accumulated in the spaces. So that is why the quantitative values of all these factors reach a steady state, and they will remain balanced at these steady state levels until something changes the rate of leakage of proteins and fluid from the blood capillaries. So coming to a summary of the functions of the lymphatic system, it is a route for absorption of nutrients, especially fats from the GIT. It plays a role in immunity. It regulates interstitial fluid protein concentration, volume and pressure. Now coming to the significance of negative interstitial fluid pressure, it acts as a means for holding the body tissues together. Traditionally, it has been assumed that the different tissues of the body are held together entirely by connective tissue fibers. However, connective tissue fibers are very weak or even absent at many places in the body, particularly at points where tissues slide over one another. Example, skin sliding over the back of the hand or over the face. Yet even at these places, the tissues are held together by negative interstitial fluid pressure, which is actually a partial vacuum. When the tissues lose their negative pressure, the fluid accumulates in the spaces, and this condition known as edema occurs. So lymphedema is nothing but collection of lymph in the interstitial tissues following the blockage of lymphatics.